started education channel, a YouTube channel which brings you about mathematics and biology. I'm teacher Chris as usual on micro. Now we are going to discuss about biology on the replication of plant virus. This is the point on the viral replication. So here, what is the replication of plant virus? How the plant virus replicate? The virus that replicate within the plant cell follows the same basic steps described earlier for the general replication of virus. So those steps that are likely to follow, there's what you call entry. Entry of plant virus into a plant often involved abrasion or insect bites. The entire virus enter the plant cell by endocytosis. So this is what we call entry. The virus are likely to enter in the plant cell through by endocytosis, taking inside of food. So here there is what we call uncoating of the plant virus nucleic then occur within the plant cell. So this process of uncoating is taking place in plant, and the nucleic acid occur within the plant cell. The spreading of the plant virus can be fulfilled by the bridge between plant cell. So this is what you call replication of plant virus, and this stage what you call the spreading of plant virus can be fulfilled by the bridge between plant cell. And you have to know that this is like a structure that is representing historical perspective. Plant virus showing the RNA can serve as the hereditary micromolecules. This is stains, then these are RNA proteins, hydric virus infections, then viral replication and stain A. This is how it takes place. So here, replication of animal virus. This is what you call the general phase process. How the replication of animal cell takes place. Animal virus, yes, takes place. The general phases in the life cycle of animal virus are the same as those of bacteria and plant, and include adsorption, penetration, and coating, synthesis, assembly, and release of from the host. These are processes that are likely to see in the process of plant viral replication, and this also takes place on the replication of animal virus. Then the length of the intermultiplication cycle varies from 8 hours in provirus to 36 hours in hypervirus. So the difference between that of the viral replication and animal replication with that of plant replication, for that of animal replication, it takes large time or large period, where it takes 80 hours. For instance, for those in animal, in plant and viral, we find that they likely to use minutes, but not in constant of the consistent of the hours. But in the animal virus, they likely to take hours, in about 80 hours in poliovirus and 36 hours in hypervirus. So also in animal replication, in animal virus replication, the animal virus exhibits some impressive mechanism for the entering host cell. So here some animal virus are uncoated outside, yes, and the flexible cell membrane of the host is penetrated by the viral nucleus. And another means of entry involves direct diffusion of the viral envelope, and with the host cell membrane, or what you call as an influenza and mumps virus. In this form of penetration, the envelope merges directly within the cell. Then after membrane, they liberating the nucleocapsid into the cell interior, and they have what you call process of penetration. If penetration takes place by endocytosis, the entire virus is engulfed by the cell and enclosed in a vacuole or a vesicle. When enzyme in the vacuole dissolve the envelope and the capsid, the virus is encoded. And the process that releases the viral nucleic acid into the cytoplasm, the exact manner of uncoating vary, but in most cases, the virus fused with the wall of the vesicles. And here, this is the process, how it takes 3 different modes of animal viral entry. Let me zoom in order to see. more about this. So try to check enveloped virus fusing with plasma membrane. This is entry of enveloped virus by endocytosis. This is what we call capsid, then spikes and the envelope. They try to enter this by engulfing. This is what the process of engulfing. If they try to engulf, they likely to be inside of the cell. This is what we call moving inside of the cell and forming nucleic acid. And if there is nucleic acid, this is what we call capsid of the virus. Then they enters in this stage. Also check as the same as this one. So let us see the next. Direct penetration by naked virus and the envelope virus fusing with the plasma membrane. This is where the envelope virus fusing with the plasma membrane. So also the entry of the envelope virus by endocytosis. This is what we call endocytosis. Then in the also this is what we call retrovirus RNA virus that use the reverse transcriptase to produce the DNA molecules within the host cell. This is the process of protein synthesis. If you remember this, try to revise on the protein synthesis. The RNA, this is RNA, this is the retrovirus injecting into the cell by endocytosis. If they reach here, they take place what we call reverse transcription, then enters into the nucleus of the cell. These capsids, though these nucleotide that released by this virus, they likely to be take place or to be inside of the cell in the nucleus, and the transcription is taking place inside of the nucleus. After this transcription, what we call RNA replication, they likely to make replication, but first there is a synthesis of the viral protein. After this synthesis of viral protein, they likely to be released out of into the cytoplasm, and then the viral resemble. So the human immunodeficiency virus, which causes AIDS, acquired immunodeficiency syndrome, is a kind of retrovirus. Here, replication of animal. You have to know that during this process, the nucleocapsid binds to the membrane, which curves completely around it and form a small pouch, pinching off the punch released to the virus within the envelope. So here, this is how the process taking place. These figures are likely to take under microscope and try to view it or to be magnified by electronic micrography of HIV binding from the host cell. This is how those HIV binding to the host cell. These host cells, these are the HIV virus. So here there is what we call, try to observe this clearly. Do you understand this? So let us see what goes on in this. Maturation and release of enveloped virus as the parain influenza virus in the binding of the membrane. So call illustration of action AZT azidotimidine in blocking HIV replication. These are the processes or different ways that are likely to be used. Plus C, phosphorylation. This is what you call process of that likely take place in the process of protein synthesis, block reverse transcription, and here we form RNA and DNA. So I think our module is at the end. I'd like to invite each and everybody who's watching this video first make subscribe. Understand the process of this. You know that this kind of virus is called oncogenic virus. The genes that induced such class transformation are called oncogenes. So these are likely to be an end of our module. Thank you for watching this video. subscribe, share, comment, and like for more about this. Thank you. I'm Teacher Chris.